Recording. Okay, Jill and Audrey, I'm recording now. I'm going to just shift this over. I hope there's a way that you'll be able to delete my control panel at the bottom here. Okay, just got rid of it. Okay, excellent. So please, hopefully the breathing isn't too heavy, and I'll go ahead. Jill or Audrey, just so you know, if I mess up a slide, then I'll just say redo or something along those lines and then count to 5. And I will pause in between slides just in case there's something that, a rustling of papers or anything that you want to delete out. So, okay, I'll start from there. So, thank you very much for tuning in for this presentation. This presentation is basically speaking about our capabilities for the Protein Passport and building the ultimate analytical testing platform. This presentation will describe in detail our approach for absolute quantification, as well as our orthogonal approach to protein analysis. Together, these two basically make up our ultimate analytical platform. The gold standard of immunological testing has played a pivotal role in the biologics area. I'm starting over. I haven't read through this in a couple days, so hold on a second. Immunological testing has played a pivotal role in early stages of biologics era and has been for many years the gold standard for detection and quantification, in particular during protein PK. However, though molecular techniques have matured and been fine-tuned, the results still yield qualitative or sometimes semi-quantitative results rather than results with absolute quantitation. The negative results of molecular assays have been confounding and complicated to troubleshoot. Oftentimes they lead to many other questions, such as, is this assay not actually optimal? Do I actually have the same molecule? That is, the molecule that you're trying to analyze for, is it the same or has it changed during the scale-up process? Did I use the correct reagents, in particular the antibody that I selected? Well, positive results are promising. Positive results still do not recognize the potential that there may have been dramatic changes within the structure, and ultimately they can lead to setbacks in the scale-up process. Obviously, immunoassays can't be replaced for functional characterization of a therapeutic or diagnostic antibodies. Nevertheless, we suggest an alternative and complementary analytical platform here where we can provide more complete and quantitative results. Here we present this alternative. I'll start over because of the paper moving. As an excellent case study for a particular case where structural analysis during the scale-up process was needed was the case of myozyme. Myozyme, which is a recombinant form of acid lysosomal glucosidase. Starting over. An excellent case, starting over. A pivotal case study can be seen here where we actually show that during the scale-up process it is necessary to be assessing the biochemical and biophysical structures along the way. Myozyme, which is a recombinant acid lysosomal glucosate, I can't say that, I'll just take that out. As an excellent example for the importance of actually testing sugar structures as well as post-translational modifications during the scale-up process, I present myozyme. Myozyme is a recombinant enzyme that's used in replacement therapy for treatment for Pompe disease. In 2006, the FDA approved this molecule under the myozyme label. The manufacturer, Genzyme, then went to scale up production from 160 liters in 2006 to 2,000-liter batches to meet the market for the enzyme therapy. However, upon scale-up, it it was first applied for approval under the Myozyme label. For many reasons, there were difficulties with respect to actually gaining approval, and the main one being that the FDA considered it substantially different based solely on its sugar structures and post-translational modifications. Gemzyme was then required to apply for a new application or a new BLA and apply for under a new brand name called Lumazyme. And during this whole process, an immunoassay was run, and it was confirming that we were having production, but wasn't actually detecting any of these differences in the sugar structures. In contrast, a targeted proteomic approach based on advanced HPLC separations of protein digest coupled with tandem mass spectrometry, or LCMS, and a protein database is free from the drawbacks that we saw before, which we spoke about before with immunoassays. Starting over. My notes are kind of messy, and I went through this several times before doing it the other night. In contrast, a targeted proteomic approach based on advanced HPLC separations, protein digest coupled with tandem mass spectrometry, as well as a protein database search, or protein fingerprinting, is free of the drawbacks that we saw in immunoassays, and in particular for protein PK. Protein quantification using this method for protein PK can be quickly achieved through easily reproducible, stable labeled representative peptides. These peptides can actually be designed and purchased once and used throughout the entire development process. Also, physical characterization and physiological stopping and restarting. In contrast, a targeted proteomic approach based on advanced HPLC separations of protein digest and protein database search, or protein fingerprinting, is free from the drawbacks and can be used to study protein PK in vivo. Protein quantification can quickly, I didn't like that I said in vivo at the end there, so I'm going to in contrast, a targeted proteomic approach based on advanced HPLC and protein database searches, or protein fingerprinting, is free from the drawbacks that we saw with immunoassays and can be used to study protein characteristics in vivo. Protein quantification can be quickly achieved through easily reproducible, stable labeled representative peptides standards, which can actually be designed once and purchased only once and used throughout the entire development process. Also, orthogonal approach to physical properties of the protein, an orthogonal analytical approach to physical properties of the protein in drug formulations such as purity, homogeneity, solubility, structural integrity, and aggregation state can actually be assessed for compatibility of the biophysical properties of the molecule. Yeah, that whole sentence came out wrong. I'm going to start over on the entire slide. In contrast, a targeted proteomic approach based on advanced HPLC and protein database search, protein fingerprinting, is free from the drawbacks that we spoke about with immunoassays. These methods can actually be used to study protein characteristics in vivo. Protein quantification can quickly be achieved through easily reproducible, stable labeled isotope representative peptide standards, which can actually be ordered only once throughout the process of the entire development. Why can I not, like, I rocked it the other day, but I also went through it several times. Okay, one more time. In contrast, our targeted proteomic approach based on advanced HPLC separations coupled with protein database search, which i.e. protein fingerprinting, is free from the drawbacks and can be used to study both protein characteristics as well as PK in vivo. Protein quantification from in vivo samples can be performed quickly through easily reproducible staple labeled representative peptide standards, which can actually be designed and purchased throughout the entire development process. Also, using an orthological approach, physical properties of the protein in the drug formulation, such as purity, homogeneity, solubility, structural integrity, aggregation state, and thermostability, can be assessed through complementary biophysical methods, constituting a biological protein passport of the protein drug. The utility of this sort of characterization is clear. Whether you're specifically developing a biosimilar for market where extensive characterization is required, improving an existing drug for extended usage, or developing a novel protein, Protein Passport can serve as a background and supporting information for a BLA or a biosimilar filing and other regulatory documents. I'm now going to go through basically our orthogonal approach. We've actually selected two model proteins to go through this approach. I will speak about its use in PK as well as general characterization of the molecules. Here we selected the top molecule, which is human growth hormone, and the lower molecule, which is interferon beta 1A. Both of these are currently available on market. So they are compounds which are clinically in use and being applied for patients, as well as both actually do have biosimilar options. PK is actually a central point for considering an analytical platform and whether or not it should be accepted or rejected. Many times in the case of PK analysis, the artifacts that are actually associated with immunoassays are most apparent and may become confounding as well as rate limiting. The specificity and selectivity of your assays is oftentimes solely reliant on your antibody and its specificity and selectivity. As the proposed alternative, we suggest using a total proteomic approach. Here's our general workflow for the total proteomic approach. It really does employ both biochemical and bioanalytical techniques, as well as using a number of proteomic databases to actually compare and select specific representative peptide markers. For large molecules, oftentimes it can be confounding to actually do absolute analysis, so here we select a representative peptide to use as a standard for comparison. I'll go through the selection process, but after we do select it, we then develop and order a standard which is actually staple labeled, so slightly heavier. We then quantify the target peptide within the samples using the heavy peptide standard as the analytical marker. We're going to go through that one more time. Here's an example of our workflow for the targeted proteomic approach. You can see that we use both protein databases as well as using analytical methods using the LTQ Orbitrap and capillary LC to actually determine the ideal target representative peptide that we will be using. For large molecules, it can also often be difficult to actually analyze the whole molecule, so here we select a representative target peptide. We then order a stable labeled peptide standard, which we then use to quantify the protein in the PK sample through absolute quantification using the internal reference standard as our, let me start over again. Here's an example of our workflow. We use both protein databases as well as analytical methods to actually select and identify a specific target peptide sequence which we will use. We then use an internal reference standard that's been stable labeled as our way to quantitate the protein in the PK samples. I'll go through this entire process more in depth in the next few slides. First, starting with protein sequence characterization using LCM-SMS. So really what we do is we actually use the digest of the protein, and then we couple this with a database search. As you can see here, here's a full scan of our digested protein, and you can see in the bottom box that we're actually getting really sufficient coverage of the entire molecule in this process. And it really begins with this protein digest and mapping. And from here we actually move into a characterization method using a database. So here we have a simulated human growth hormone digestion, and we actually can select detect specific peptides that we can then cross-compare back to our already digested and analyzed sample. There's a number of peptides which we may pick, and they're selected based on specific sequences as well as the residues and the properties that the residues actually have. And then we compare these back to the digested analyzed sample actually to look at abundance. At that point, these two factors will actually be used to determine which of these peptide sequences may be the ideal sequence. We then move forward with considering potentially, we go from several potential peptide sequences, and we can narrow it down to the top 5 as we've done here. For future, in the up and coming slides, we're actually going to look more specifically at our peptide sequence SNLELLR, and this was selected as one of the main peptide sequences that we would look at based on the abundance that we found in the in vivo sequence, in the in vivo digest, which we got through the analysis, as well as based on the database search. From here, we can then also compare what the feasibility of the methods would be for development. So specific peptides may actually be limited based on the method development process, which I'll again show later. Once we've actually decided on the specific protein sequence that we'll use, we can actually go ahead and order stable labeled peptides, which are available through a number of vendors. Upon receipt of the peptide sequence which we have ordered, we always perform a representative tandem MS spectrum of our specific peptide sequence to confirm that the peptide that we received is the one that we ordered. Additionally, as part of the method development process, it is important to show that, starting over. As part of the feasibility of using this peptide mapping process as an excellent model to do absolute quantification in vivo, we did want to show that the analytical sensitivity was such that you could reach a dynamic range, dynamic concentration range, and still detect specific peptides. As you can see here, peptides can actually be resolved over a range of concentrations. And depending on the compound itself and how much we anticipate there being in the PK samples, we can actually develop models to achieve more relevant lower limits of quantification. In addition to looking at the dynamic range over concentrations for the analysis purposes, we also used a total proteomic approach using both the digest as well as using protein databases to show what the coverage is over a dynamic range of concentrations. As you can see, with 1 microgram per microliter, we have about 97% coverage in this high concentration. As we reduce that by 100th, we still retain about 80% coverage within the molecule itself. You can actually see, specifically looking at our SNLELLR sequence, we have good coverage up here, and the good coverage continues down into our lower limit, our lowest limit that we actually went to here. Now, all the previous data that we've actually shown in full scan was in an in vitro test system, that is to say, a physiological buffer. However, there are challenges specifically for protein PK, and one of the main challenges is whether or not the protein that's actually in the blood serum that you're harvesting. So the concentrations that we're actually achieving in vivo can be much lower than, let me start over. To this point, the sequence-specific characterization that we've demonstrated have been in vitro test systems, also at microgram level. That is to say, it was in a buffer system at a fairly high concentration level. And this is represented a feasible task. But where the additional challenges are for protein characterization and quantification in vivo as opposed to an in vitro system lie in the concentration that we can achieve as well as the proteins that are actually in the matrix. For human growth hormone, the data on the left here shows the levels of protein after a single dose of in humans of 0.01 to 0.1 micrograms per milliliter as opposed to micrograms per microliter, which we had previously looked at. This is less than the levels that we've actually demonstrated in our previous slides. However, this sensitivity can be achieved through replacement of analytical size HPLC column with a micropore column, or even using a nano LC system. Another challenge is actually the resolution in the representative matrix. The resolution of the specific representative peptide may be difficult because of the complex mixture of peptides and proteins that are actually in the biological matrix, in this case serum. As you can see in serum, it's about 70 mg per mil of protein, and about 80% of that total mass is actually made up of albumin and IgGs. Unfortunately, using the targeted proteomic analysis based on LCMS, we can actually overcome these samples. I'm going to repeat that last sentence. Hopefully you can just switch it out. Fortunately, based on proteomic analysis, actually I'll just repeat the whole slide because that's easier. To this point, all the sequence-specific characterization of protein that we've actually shown in this presentation has been an in vitro matrix, that is, a buffer matrix, at microgram levels. And this has shown that this is a feasible task. But what challenges actually lie for protein characterization and quantification in vivo? Really, we've noted that there could be two distinct challenges here. First, as you can see in the left panel, For human growth hormone, the data show that the levels of protein after a single dose in humans is actually 0.01 and 0.1 micrograms per mil. Now, our previous data was actually using micrograms per microliter, so you can see that this is a significantly lower concentration than we had previously detected. However, we do know that we can actually achieve better sensitivity for this method through replacement of the analytical size HPLC column with a micropore, or even using a nano-LC system. The other challenge we do realize is that the resolution of the specific representative peptide may actually be much more complex when using a specific biological matrix. Here we're using blood serum as most PK samples will use, and you can see that blood serum is actually about 70 mg per mil protein, and 80% of that total mass consists of albumin as well as IgG. Fortunately, the targeted proteomic approach based on LCMS can actually overcome these problems. First, starting with the quenching of the signal in a biological matrix. For all the subsequent studies that I'll be going through here, we're actually using the lowest concentration that we had used in the in vitro matrix, or the buffer system, at 0.01 micrograms per microliter. In the top figure, or in the top scan, we can actually see this is the 0.01 micrograms per microliter spiked into a buffer system. And you can see distinct peaks, which we're actually going to detect. However, in the middle scan, we actually show what it looks like once we do to the protein digest when the specific protein that we're looking for is actually spiked into 100 micrograms of serum. And you can see after the protein digest, even the 100 micrograms of serum completely mask any sort of peptides which we may be getting. However, one way that we identify to actually reduce this background is actually through depletion of serum. So as you can see, doing a depletion of human albumin as well as IgG, we actually have significant reduction in the background as well as an increase in the resolution of specific peptide peaks. In addition, with and without depletion, we can see that we go from about 80% coverage here, reduced in the presence of serum to about 28% coverage. So we have very low levels of peaks that are specific to our actual human growth hormone, very low coverage. However, with a simple depletion of the human serum albumin as well as the IgG, you can see that we actually have an increased coverage to about 47%. Further depletion of different serum peptides, as we know, and we have identified about 20 different peptides which we actually can move to deplete, can actually lead to better resolution. Additionally, one must consider that the representative peptide that they're using may look great in an in vitro matrix like a buffer. However, it is important to do additional method development in the specific matrix that you're going to be looking at. Once again, here looking at our representative peptide SNLELLR, we can see that in the buffer system we get a very strong peak. However, in the presence of serum, following aligned with the similar studies that we've done previously where we have serum and then deplete serum, in the presence of serum you can see that it really masks the strength of that, the peak that we're actually looking at, and reduces the resolution of that peak. However, after depletion, we've practically cleaned up the contaminating peptide ions which are present because of the serum peptides and proteins. So you can see that in this case, the use of SNLELLR would be sufficient, and additional work can be done to achieve relevant lower limits of quantification. However, looking at peptide LEDGSPR, which we had identified as a promising representative peptide in an in vitro matrix, we've actually then gone to spike it in, and you can see that once again the background now completely wipes out that detection of that sharp peak. However, after depletion, once again, we basically don't have a very strong identifying peak due to background proteins and peptides. So in addition to using the different requirements we actually had for identifying the peptides, that is to say abundance as well as specific properties of the protein of the amino acid residues, we can also use this as an additional criteria for selection of our representative peptides, which will be used as our internal standards. If we're unable to resolve the peptides in our relevant matrix, perhaps this peptide shouldn't be used. However, as I did mention before, there are a number of methods that can be used to increase sensitivity and resolution if you're interested in using a specific peptide sequence, and that would be using a higher level of depletion, that is including other peptides, so there's up to 20 major peptides which can be removed, or increasing the resolving power using different HPLC gradients. We've actually continued work on these projects, and I would be happy to speak with you more individually, but in consideration of time, we will be moving forward to speaking about our other orthogonal approaches. However, if you are interested in this, please feel free to contact us. So in addition to the absolute quantification of proteins, as I mentioned, we take a full orthogonal approach to actually protein analysis. And post-translational modifications are really just as important to characterization of a protein or a peptide as I would say the importance of being able to do absolute quantification of proteins in PK samples. So here we look at using our high-resolution accurate mass spectrometry, once again, which we had used for all the previous studies, to actually analyze the post-translational modifications. Here we're specifically looking at the glycosylation of interferon beta 1a using high-resolution accurate mass spectrometry. Obviously, we are specifically looking at glycosylations, but these methods can actually be applied to a number of other post-translational modifications as well. So here's the general mass spectrum for human recombinant interferon beta 1a, and you can see that we've demonstrated the presence of several isoforms at different masses. Using a database approach, using glycodatabase, we actually can analyze for the presence of the above carbohydrate structures, which actually differ within branching, fucosylation, and sialation content. The advantage of this approach is the opportunity for relative quantification of different glycan forms. So we can see that we have a number of different glycan forms present, and we can actually do relative quantification of all of them. The specific applications, such as we can also use it for specific applications, such as analysis of glycosylation sites, LCMS, protein digest can actually, I'm going to start this slide over because I screwed myself up. Here we show the glycosylation of interferon beta 1a using our HRAMS, that is high resolution accurate mass spectrometry. The mass spectrum above we can actually see demonstrates that there's several isoforms with different masses present within one sample. Using a database as well as our analysis, we can actually see that it suggests that the above carbohydrate structures, which may actually differ within branching of their carbohydrate structures, fructosylation as well as sialation content. The advantage of using this approach as opposed to different approaches is that you can actually see the relative abundance and relative quantitation of the different glycan forms within a sample. For other specific applications, such as analysis of glycosylation sites, LC-MS of the protein digest in combination with carbohydrate trimming enzymes can be suggested. But obviously, depending on the protein and the project, specific method developments may be required. However, the general analytical platform can be suggested for any post-translational modification or any protein that has post-translational modifications. Continuing with our orthogonal approach to analysis and stability characterization, we can see that through a number of biophysical and biochemical techniques, you can actually confirm that the protein is folding. Oftentimes, this can be used as is folding correctly. Oftentimes, this can actually be used as a litmus test for scale-up process. So by looking at protein from the primary, secondary, tertiary, and potentially quaternary level, we can actually see that we do have the correct protein. So that is to say, the protein that you started out with is the protein that you're ending with. So in this case, we can be looking at the secondary as performed by the circular dichroism, where we can look at beta sheets and alpha helices, or we can use differential scanning calorimetry to look at melting curves, as well as intrinsic fluorescence to see how to see the fluorescence of the molecule itself. And all of these can be used, as I said, as a passport to actually moving on to the next step. So during the scale-up process, or going from your in vitro studies, which have been shown promising results, to your in vivo studies where you may need significantly more protein, you can actually use these as a litmus test as to whether or not batches can be approved and released for use. In addition to this, it is always important to show purity, homogeneity, and to test aggregation. So using dynamic light scattering as well as size exclusion chromatography, we can actually look at the purity, homogeneity, and aggregation. Purity and homogeneity obviously are very important because if you have an unpure protein sample or a heterogeneous protein population which you're going to be using, and there are any adverse side effects, the question is always, well, was it our protein that's most present there, or was it the few artifacts that I may have from the overall biotechnology process? Obviously, these impurities can also lead to negative side effects, such as immunogenicity in moving into the actual in vivo studies. Secondly, you can also look at aggregation. Aggregation of the molecule is are basically when the molecules are all coming together, and this can absolutely lead to immunogenicity issues. So these sorts of methods using dynamic light scattering as well as size exclusion chromatography give you the confidence that the molecule and the lot of molecule that you're moving forward with your studies is pure, homogenic, as well as your risk of aggregation and immunogenicity is minimalized. As we spoke about, instability or potentially unfolding of a native peptide can actually lead to reduction in activity. So the native peptide, as it's normally found in nature or in your body or in a plant, if it's a plant or plant-derived molecule, has high activity in its natural state. However, oftentimes when putting this into an artificial state, let's say in an artificial buffer, which we're then going to be using as a for the dosing into animals and humans, or even if you're making a human recombinant, can lead to some chemical and physical instability. The chemical and physical instability may be reversed. However, sometimes this may lead to intermediates, which can then aggregate and ultimately precipitate out. The precipitation out, as well as the aggregation, if you're not aware of it, and it may be something you may not be able to see with the naked eye, may actually lead to aggregation and adverse side effects in your preclinical studies. One way to actually alleviate this potential of instability, if you're worried about the instability, is actually through formulation. Selection of the correct excipients and formulation buffers as well as pHs may allow to keep your compound within that native state and achieve maximum activity in your studies. Finally, much like instability leading to issues with both the types of buffers that you can actually use as well as the types of excipients, solubility may actually have a constraint on the product profile itself. The product profile can be defined as, the route of administration, or part of the, I'm starting over. Much like instability can actually put constraints on development of molecule, solubility can also put constraints on your product profile. The product profile really defines the method with which you're actually going to be dosing the molecule. So in this case, it's an excellent case study where we worked with a company who was looking to reformulate because the solubility of the molecule put a constraint and a strain on the method that the dosing could actually take place. So in the original formulation, which they had determined, based on the solubility constraint, which was about 60 mg per ml in a mannitol-based formulation, to achieve the relevant concentrations and the relevant active concentrations, it actually required patients to come in once or twice a week for a 4-hour IV infusion. Now, obviously this isn't necessarily patient-friendly. So our customer came to us looking to actually target a different dose route, which was much more patient-friendly. So they were opting for an intramuscular or subcutaneous route that the patient may actually be able to deliver to themselves. And even if they couldn't deliver it to themselves, it would only require potentially a 15-minute doctor's visit. So with this in mind, our target or our product profile actually defined the solubility that we needed to achieve. And the solubility that they desired to achieve from a 16 mg per ml solubility in a mannitol-based formulation, they actually wanted to achieve about 100 to 150 mg per ml. This was completely impossible using the mannitol-based formulation. So what we did was we were able to look at the type of biologic it was itself and come up with potentials, different formulations which would be able to increase the solubility of the molecule. And our success was that using an amino acid based formulation, we were actually able to achieve a relevant the concentration well above what they actually were targeting, which was 150 mg per ml, and we allowed for about 250 mg per ml, and hopefully, potentially, eventually, their target dose route of intramuscular and subcutaneous. So this reformulation actually helped our customer to determine a no-go, go decision on whether or not to allocate resources towards this reformulation. And we do anticipate that for 2012 we will be doing a lot more formulation work for this customer. So though biological activity and immunoassays still play a very pivotal role in the process of developing a biologic, it's clear that it's not just about the immunoassays. A focused approach using a focused approach, I'm going to start over. Though biological activity and immunoassays still play a pivotal role in the development of and analytics of a biologic, it's clear that using a more quantitative approach may also be useful. Analyzing the protein from the bottom up, from primary, secondary, quaternary, and potentially tertiary levels really helps to get a full picture of the molecule. Whether you're using this protein passport during the scale-up process, when you're going from your innovator molecule, which showed great efficacy in vitro, scaling up into your preclinical species and ultimately your clinical species, or if you're using it to analyze your PK samples through the total proteomic approach, which I had previously described, it's clear that the approach must be both a mix of our steadfast immunoassays as well as the new developments in our analytical capabilities. I hope that I've impressed upon you the importance of looking at different facets of analysis and looking at the protein. I'm hoping that you can just edit out that last section. So I'll start with the weather. I hope. I hope that I've impressed upon you the importance of looking at both the immunoassay portion of analytical development as well as using the strict LC-MS mass spectrometry analytical development and showing you the ultimate analytical testing platform for proteins. Please, if you have any additional questions or specific questions on how this would apply to your protein, please feel free to contact us. We'd be happy to get in touch, and we'd be happy to speak more specifically about the development of and application of these processes to your protein. Thank you very much for your interest, and I hope to hear from you soon enough.